Immune responses are not always protective in nature but can be associated with tissue destruction and disease. It's now evidence that pathological immune processes in autoimmune disease use immune cells and mediators similar to those in protective immune action. Fundamental properties of the normal immune response, as we know: antigen-specific recognition, diversity of the lymphocyte repertoire, discrimination of self versus non-self, immunological memory, self-limitation. The ability of T cells to respond to an antigen depends on the availability of the correct antigen receptors. It's estimated that about a possible TCR are available in our bodies. This level of diversity can be generated by combination of different mechanisms as multiple germinal theory, exon rearrangement or combination diversity, junctional diversity and junctional flexibility. These gene segments are often translated in all three reading frames: alpha and beta chain association. These TCR, beta and alpha chain gene recombination and expression, the germline DNA, rearranged DNA, and the primary DNA transcript, the messenger RNA, and nascent polypeptide TCR chain assembled TCR molecule. By definition, specific controlled inability to respond to antigen to which an individual has the potential for response, elimination or neutralization of the autoreactive B and T cells. A breakdown of this working system can cause physiologically. It's directed against antigen from the normal tissue, but it can be induced against any antigen by the use of appropriate procedures such as administration of antigen to very immature animals, administration of very large or very small dose of antigens, initiation of antigen treated in such a way that it could not be processed properly. When a specific lymphocyte encounter antigen 3, possible outcome may occur. The lymphocytes are activated, leading to immune response. The lymphocytes are inactivated or eliminated, leading to tolerance. The antigen is ignored. The choice between lymphocyte activation, tolerance, or ignorance depends on the nature of the antigen-specific T lymphocytes, nature of inducing antigens, nature of presentation to immune system, activation of naive T cell in the presence or absence of co-stimulatory signal, an overview of the lymphocyte response, the viral resistance, infected cells, cytotoxic T cells, and this is the macrophage and the mesohistochondrial presenting T helper cells, virus, interferon, interleukin, interferon gamma, interferon 2. This is the infected cell and the virus. Follicular dendritic cell uptake the microbe of microbial component. We discuss this in here. The normal immune response, immunogenic antigen, proliferation and differentiation. Here the normal immune response, the tolerance, tolerogenic antigen, energy, functional unresponsiveness or apoptosis, cell death, immunogenic antigen, no response, ignorance, non-immunogenic antigen, immunogenic antigen. This is the fate of lymphocyte after encountered with antigens. The importance of tolerance. Self-antigen normally induced tolerance. If we know how to induce tolerance in lymphocytes specific for a particular antigen, we may be able to use this knowledge to prevent or treat unwanted immune response. Self-tolerance phenomena. It's a tolerance to self-antigen which is characterized by it's not inherited, it's antigen specific as in immune recognition. It's achieved through clonal deletion of the self-reactive lymphocyte through clonal energy of potentially autoreactive T and B cells. It may be overcome, leading to autoaggressive immune recognition. General feature of tolerance: dose dependent. Large or small dose of antigen are potent inducer of the tolerance. Site dependent antigens ingested orally are more likely to induce tolerance. Delivery dependent antigen in the absence of co-signal result in energy. mechanisms of tolerance: central tolerance, immune self-reactive lymphocyte, self-antigen; peripheral tolerance, mature self-reactive lymphocyte, self-antigen. Some self-antigen are ignored by immune. Mechanism of tolerance induction in T cells: central tolerance, positive selection, negative selection; peripheral tolerance, regulation, energy and inhibition; deletion, activation, induced cell death; immune suppression, ignorance, immune paralysis. Central tolerance. Central mechanism of the induction of tolerance: step CD80, CD24, antigen presenting cell, and here this signal and apoptosis occur, or another pathway, maturation and surviving of the cell and leave the thymus. The central mechanism of induction of toleration, it's the selection of T cell carrying receptor which can recognize the self MHC antigen. This process occurs in the cortex of the thymus. If the thymocyte recognize self MHC with intermediate affinity, they will be stimulated to proliferate into colonal expansion. If thymic epithelial cell present antigen in association with MHC class 2, the T cell will lose CD8, change to single positive CD4 T cells, but if they recognize MHC class 1, they will be signal positive CD. The positive and the negative selection in thymus, the thymus gland. This is occur inside the thymus gland. Negative selection, T cells with receptor binding with high affinity to the self-MHC self-peptide complex will be eliminated either by clonal deletion through induction of apoptosis, clonal energy by inhibition of interleukin 2 production. Negative selection will ensure the removal of self-reactive T cell, leaving only two T cells which are self-tolerant, MHC restricted, reactive to foreign antigens. Normal response and clonal energy, T cell energy, and antigen presented by a co-stimulatory expanding antigen presenting cell. A normal cell response: if the T cell recognize antigen without co-stimulatory or in presence of CTA A4 B7 interaction, the T cell fail to respond and rendered incapable of responding even if the antigen is subsequently represented by co-stimulatory expressing antigen presenting cells. The normal response, antigen recognition. As we know, antigen recognition will with co-stimulation, the T cell response, T cell proliferate and differentiate. Co-stimulatory deficient antigen presenting cell restimulation with antigen presenting cell expressing co-stimulation, co-stimulator T cell energy. The T cell proliferate and differentiate. Here in this pathway, altered peptide ligand and failure to respond to native peripheral tolerance. There are two signals required of T cell activation. The first signal is from the interaction between TCR and the peptide antigen. The second signal is from the interaction of co-stimulatory molecule CD28 T cell and the CD B7. The relation between CD28 and CD4. Mechanism of peripheral T cell tolerance: ignorance, deletion, regulation, energy and inhibition, suppression and deviation. Immune ignorance: self-reactive lymphocyte may fail to recognize or respond to some self-antigen in the periphery, but the cell neither die nor become energetic. Mechanism of ignorance may be the level of antigen may be below the threshold required to induce the activation or deletion of T cells. The antigen may be physically separated from T cells. The antigen may be presented in the absence of co-stimulation, absence of T cell help, deletion, repeated activation of mature T lymphocyte by self-antigen, recognition of self-antigen without second trigger pathways of apoptosis that result in elimination of the self-reactive lymphocyte. This process called activation-induced cell death. Mechanism of activation-induced cell death: co-expression of the death receptor called FAS, CD95 and its ligand FAS ligand. Lack of an excess of the growth factor, especially interleukin-2, may stimulate or suppress immune response. Antigen recognition by T cell induces the production of pro-apoptotic protein, which normally antagonized by anti-apoptotic protein produced during immune response produced by microbe and not produced in case of self-antigen recognition. The normal response: antigen recognition, T cell response, activation induced death on T lymphocyte. Here apoptosis, expression of the FAS and FAS ligand, and this the apoptosis. Regulation, energy inhibition. Energy: T cells that don't reproduce interleukin-2 on encountering their antigen and that therefore can't be completely activated are called anergic. Energy may have widespread consequence because certain anergic T cells produce interleukin-10, which suppress the activation of T cells. No specific phenomic marker of energy have yet been discovered. Inhibition, CD152, also referred to as cytotoxic T lymphocyte associated protein on T cells, bind CD8 and CD86 on B cells with higher affinity than the co-stimulatory receptor CD28. By this way, CD152 inhibit the activation of T cells. The function of CD28, B7, B72, and CTLA molecules. The interaction between these: here inhibition, here unresponsiveness, co-stimulation, the enzyme burns the cell, the cell defeat the T cell, the antigen presenting cell, and the T cell, and CD28 cell separation. Some self-reaction to T lymphocyte may develop into regulatory cell whose function is to prevent and suppress the activation of other potential harmful self-reactive lymphocytes. Regulation of T cell may develop in thymus or in the peripheral lymphoid organ. Most regulatory T lymphocytes are CD4 and express high level of CD25. The feature of antigen-derived T cell to be suppressor and not effector, and the mechanism by which T cell inhibit immune response are slightly known. However, there are several ways in which T cell may suppress the function of the another cells. Soluble inhibitory factors, the civil servant model, soluble inhibitory factor, cytotoxic separation, idiotype mediated separation, inhibition of T cell activation, inhibition of the T cell effector function, normal response and Hazelden separation. Here the normal response: naive T cells activated. Here the interferon gamma activated macrophage, interleukin 10 inhibit function of the antigen presenting. Here suppression is the process of suppression. Immune paralysis: it's considered a type of clonal anergy resulting from a very high dose of antigen which bypass antigen presenting cells, reaches the helper T cell directly, making them anergic. Character of T cell tolerance: need low dose of tolerizing antigen, persistence of tolerant state of long term. It may be central or peripheral. B cell tolerance character: needs a high dose of antigen, persist for short time, can be induced either in bone marrow or periphery. Mechanism may be by deletion or anergy. Inappropriate antigen presentation will lead to failure of CD4 to deliver help to B lymphocyte. Leading mechanism: several mechanisms available to filter autoreactive B cell out of B cell repertoire. Clonal deletion of immune B cell in the bone marrow, central tolerance, receptor editing, peripheral tolerance, clonal exhaustion, blockage of PCR, lack of help from T cell, clonal energy, functional inactivation, the deletion of autoreactive B cell in the T cell zone of the spleen or lymph node, central tolerance negative selection, the immature B cell, apoptosis, mature B cell, receptor editing expression of new antigen receptor, failure of central tolerance of B cells, and maturation of the B cell to peripheral lymphocyte cell, negative selection and receptor editing in immature B lymphocyte. The mature B lymphocyte, energy block in antigen receptor induced signal, exclusion of the B cell from the lymphoid follicle, clonal exhaustion, repeated antigenic stimulation of B cells will convert the B cell into plasma cell. If all B cells are differentiated into plasma cell, no memory cell will be available for secondary immune response leading to tolerance. Blockage of BCR, some polymorphic antigens such as pneumococcal polysaccharide can bind irreversibly to B cell receptor and block any further response by these cells. Duration of tolerance dependent, resistance of the tolerizing antigen, the ability of bone marrow to generate fresh antigen sensitive cells, and the factors of the favor simulation of the immune response factor that favor tolerance, the amount, the persistence portal of entry, presence of adjuvant, properties of the antigen presenting cell. Here the amount, optimal doses, here high dose, fever tolerance, persistence, prolonged, repeated, short-lived, port of entry, subcutaneous, intravenous or oral, presence of adjuvant, antigen with adjuvant stimulate helper T cell, antigen without adjuvant, non-immunogenic, properties of the antigen presenting cell, high level of co-stimulatory, here low level of co-stimulatory. Self-tolerance in T and B lymphocyte. This difference, principle of site tolerance, T lymphocyte and B lymphocyte. Principal site of tolerance, thymus and periphery, bone marrow and periphery. Tolerance sensitive stage of maturation, double positive thymocytes, immature IgM, IgT. Stimuli for tolerance induction, central high affinity recognition of the antigen, peripheral lack of co-stimulatory or repeated stimulation, central in B cells recognition of the multivalent antigen in bone marrow, peripheral antigen recognition without T cell helper, principal mechanism of tolerance, central clonal deletion, peripheral energy activation induced cell death and suppression, central in B clonal deletion, peripheral block in signal transduction and failure to enter lymphoid follicle. Definition